Yes, in the interest of keeping to time, I will quickly pass on to Frank Vollmer for the next talk.
Yeah, thanks. And I think I have have Daniel to present the slides, so that's great. I see them already. I trust everybody else
Yep,
sees
let
them too.
me. Yep.
Okay, so thanks, Daniel. So very glad to introduce this molecular mechanics initiative or the molecular mechanics project of enzymes here in Exeter. And that will be presented by Daniel Mitchell, who you know. He's one of the organizers, and here I want to also thank all the others organizers for putting this together. And other presenters in the next 45 minutes are Nikita and Jesus, all postdocs, interdisciplinary, coming from biochemistry, physics, and theoretical physics. So next slide, please. So our project evolves around doing exactly what you see here, and that is using a golden sensor or a gold nano sensor to study record signals from enzymes, well, protein nanomachines, if you wish. We want to record signals from when they move, their conformational changes, which are often part of catalytic activity. And we want to record these signals, understand what forces drive them, and also apply optical forces to direct synthetic activities at the scale of the molecule of the enzyme. So next slide, please. Here you see the sensing concept. So the key element of the sensor is a plasmonic nanoparticle, about 80 by 20 nanometers, so about the size of these particulates that we heard about. And this particle, turns out, focus is plasmonic, and so in other words, focuses the light down to length scale of the protein, shown here, that we're going to detect because it overlaps with this concentrated light field. And fortuitously, plasmonics focuses light down to 10 nanometers. But it's not only we can detect the protein, also when it now changes its structure, meaning bonds and atoms moves, then that tunes our sensor signal. And the sensor signal, next slide, is actually a change of color. If you wonder now how does that work? Well, it's an interferometer. So of course, interferometer measures wavelengths, right? Color signal is shown on the right, a very sharp spectral feature that changes. We track it over time. You see it in the middle figure here over time, right? There's some kind of double peak that's associated actually with this hinge bending movement of this enzyme that you see here. It's like a Pac-Man immobilized on the sensor. It's moving, right? Chewing up the substrate molecules, and we get this double peak signal each time it does that. And that shows you how we study the motion. And in next slide, and in the progress of this grant, we also not only want to record signals, but also next slide of the slide. The enzyme is in these plasmonic hotspots where it is exposed to field gradients that we can control, or in other words, exert optical forces. And we don't need much here. We think femtonewtons to either oppose or apply a force in direction of the movement. So in other words, manipulate the enzyme movements and with that catalysis. And of course, we would see that next slide in our next one in our signal, because over time, right, we get these blips for principal movements, right, different patterns for different principal movements. And now as we apply a force, also, with a certain protocol over time, these movements will change, and we'll see that, and we'll know hopefully how large the forces are. So we're talking femtonewtons, difficult to measure with AFM or other traditional optical tweezers, and we may be able to direct synthesis for the first time, basically by maybe directing a polymerase to make DNA. So with that, over to, I guess, our first postdoc presentation by Daniel.
Yep, thank you, Frank, for that excellent introduction. So as Frank alluded to, our mission in our project is to measure the catalytic activity of one single enzyme at a time. So the actual activity of an enzyme as it turns over one at a time. And we're using a novel technique, as Frank previously described it, as called whispering gallery mode. This is just a fun fact. So if you go to St. Paul's Cathedral in London and you go to the whispering gallery, person standing at one side can whisper, and due to this acoustic internal reflection, a listener standing right around the other side can actually hear you whisper. So go there with a friend and try this one out. In our case, we're using light instead of sound, so we want a protein to whisper and then pick up its activity, kind of in this sphere type method. So exploiting precise measurements of optical resonance frequencies to detect very small changes. And basically you want the light to enter the sphere. You get a self-interference and an optical resonance. And if you stick something to the sphere, then effectively increases the circumference of the sphere and thus the resonance, and thus you can detect very small changes in the wavelength and very small things happening. And we use a plasmonic gold nanoparticle as effectively an antennae, which can concentrate down the light field to enable us to detect single proteins at a time. So this is sort of an overview of our setup. So we bind the protein onto a gold nanoparticle, attach the nanoparticle onto our sensor, and then hopefully we get a signal of the protein going from conformation 1 to conformational 2. And at Exeter, we have a lot of different specialities. So we biochemists, so producing the enzymes we want to look at. We have Misha, who's a very good crystallographer to get crystal structures to kind of try and figure out what's happening on the sensor, and give us a bit of modeling information. Got biophysics, so how do we actually immobilize the enzymes onto the gold and keep them active? Optical physics, so Frank's group, how do we actually build one of these sensors? And then we got some theoretical physics. So once we've got these signals, how do we actually make sense of them? And then as a sort of applications-based system, the idea is to take our sensor and use it for very high sensitivity detection of fungal pathogens. So the process, so we want to immobilize our enzyme, or first choose an enzyme and then immobilize it, build our setup, detect a signal, and then use statistical theory, structural biology, so X-ray crystallography, and enzyme kinetics to actually figure out what these signals mean. And it's sort of a cyclic process, so gradually refining each of the methods to improve our system so that we can sort of clearly figure out how to do each step successfully and work out what the enzyme's actually doing on the sensor. So for the first section, which is mainly my area of expertise, I'm going to give you a quick rundown on which enzymes and how do we immobilize them. So the first question we want to ask ourselves is, what sort of enzyme do we want to use? So I guess the key take-home message is it's got to be active when immobilized, which is, I know there's a lot of literature on this, but it's not always as easy as some of the papers make out. Also, it's good to have, we want crystal structures, maybe for a first test enzyme, a very large conformational change to make it easy to detect. And sort of as more of an interest thing, why do we want to detect this enzyme? Why is this interesting? So maybe an interesting mechanism, use in a biosensor, so for us mycology, or potentially in industrial catalysis as well, which we're also interested in. So our two enzymes that I'm going to talk about today are this 3-phosphoglycerate kinase. So this should show hinge bending. So if you think the bit in the middle where the pointer is, that acts as a hinge, and protein can flex around here, sort of like a crocodile's mouth effectively, and you get a large movement, and this should be hopefully be easier to detect. And we also have crystal structures. Simona, a talented PhD student in our project, is currently getting a load of different crystal structures at all the different intermediate stages, and so we should hopefully have the sort of the full range of different intermediate steps, and we can hopefully correspond this to any signal we get. And as a second sort of more biosensory application, we'll quickly look at this XOG1 from Candida, which is a fungal pathogen. So possibly a drug target, and it also binds this human antimicrobial peptide, LL37. So it's very interesting for biosensing applications. So we'll start with PGK as our test enzyme for its big movement. So firstly, how do we immobilize our enzyme onto the gold? So using a technique already used very successfully in protein purification. So when you express and purify a protein, you express it with 6 histidines, usually on one end of the protein, and histidine binds very strongly to nickel NTA. It's basically this molecule. So if you coat a gold nanoparticle in nickel NTA, then you should very strongly be able to bind your protein of interest onto the gold surface. So as a bulk assay to check everything works, so this is mostly done by Simona. So she did some bulk assays with the gold nanoparticles, and looking at the relative activity, we took the gold, immobilized the enzyme, and then washed it several times. So wash 1, wash 2, wash 3, and then by wash 4, there's no activity whatsoever. And then with the nanorods and the enzyme immobilized, you still get 20% of the activity, showing that a good amount of protein is bound and is still active. So we're ready to go from a bulk assay to a single molecule assay. So first we bind our nanoparticles onto the sensor, so this bit. And as you can see on this bit of the slide, so these increases are actually a single nanorod binding to the sensor, so you can detect one at a time. So we can at least bind nanorods. Step 2, adding the nickel NTA. So we add the nickel NTA, incubate it, and then again you get little steps of the NTA binding one at a time. So again, we can detect single things binding to the sensor. And then thirdly, the third step, attaching the enzyme to the sensor. So again, on the right-hand side, this nice step here. So this is a single enzyme binding through the 6 histidines on its N terminus of the protein. So we can detect single enzyme binding again, which is really nice. And now for the sort of the final money step, can we actually detect the enzyme turning over? Yes, indeed we can, which is really, really cool. So with this enzyme, you get this characteristic double peak on the addition of substrate. So we can actually detect one enzyme turning over at a time, and as it turns over, you get this movement of the enzyme, which our sensor is actually sensitive enough to pick up, and you get this nice double peak. And Jesus is going to talk a bit later about decoding these, what these double peaks actually mean and what information we can try and figure out from this. So our test enzyme is perfect and this works really nicely. So can we start to expand this to keep the mycologists happy and see if we can actually start creating a biosensor? So we'll use this exerg1 enzyme, which uses this laminarin, which is found, among other things, in the Candida cell wall. So the enzyme chops off so laminarin is a polymeric sugar, and the exerg1 comes, binds to the sugar, and then chops off the end glucose. And this is very important for hiding the Candida from our immune system. So our immune system recognizes beta-glucans really strongly. So the Candida doesn't obviously want this. So it uses exerg1 to hide these sugars. And so if we can detect exerg1, then we can hopefully detect Candida. And this would be really nice at very low concentrations. So because we want to actually detect the exerg1 in a solution, not the substrate this time, we're going to start with attaching the substrate onto the gold. So this laminarin polymeric sugar, we thiolate it, so add a sulfur and then attach it to the nanorod in this method. Interestingly, exerg1, rather than having a binding site, has a binding groove effectively, so it can bind up to 5 sugars in one go, and then this chop off the end one. So we should also see binding, and the enzyme turnover is also really slow. So this should help us both see binding and turnover. So in bulk, we can mobilize it. So the activity still kept, which is good. And you can also track immobilization using UV-vis spectroscopy also. So it's definitely immobilized. It's definitely still active on gold, in bulk at least. So we're ready to attach this one to our sensor as well. So as I said previously, we're going to start with binding the substrate. So we get these binding steps. So as one laminarin substrate binds, we can detect this. So that's very good. And then as our sort of biosensing step, once we add the exerg1 enzyme, we, in this case, instead of the double peak we got with the PGK enzyme, we have a mixture of spikes and steps. So as I alluded to, I imagine this is more of a, I guess you would more see binding because the enzyme turnover is a lot slower rather than a characteristic turnover event. But it's very interesting that we can detect exerg1. So 150 nanomolar is kind of the maximum we need to go to. So we can push this down to sort of 10 nanomolar and still actually start detecting things. So this is really promising for a biosensor application. As I mentioned on a previous slide, this LL37 peptide is also an interesting binding, has an interesting binding interaction with exerg1. So there's a couple of papers on this now, which is really interesting. So we thought, why not take this LL37, bind it onto the sensor, and then see if we can add XG1 and detect binding. And then if you add substrate, then you could hopefully also detect activity as well. Again, we're using a cysteine. So we had this peptide synthesized, so the classic LL37, but with a cysteine on the front. And again, in our bulk assay, so we added the LL37, immobilized it, and then added XG1, and then washed off all the rest of the non-bound XG1, and you actually get a massive amount of activity from bound XG1. So this seems to work as a detection method, at least in bulk. So moving on to the sensor. So LL37 binds quite nicely through the cysteine, and then adding in the XG1. So in this little schematic, so the LL37 is bound, add the protein, bind that onto the LL37, and then this seems to work. You get these binding steps, and then sort of as the final thing, you can add the substrate, and then you get either these interacting turnover steps or you get binding steps. So again, we can use this as a very specific, highly sensitive biosensor, so down to nanomolar ranges for this to, I guess, detect XG1, and also in this case, once you add the substrate, you should be able to see these nice interacting of the XG1 and the substrate. So hopefully, as a sort of further step, hopefully we can do some powerful analysis on this and hopefully pick up a unique interacting spectra for XG1 and start using this as an actual really specific sensitive biosensor. So the take-home message for this part, we now have two different methods of detecting XG1 right down to a single molecule level. So to conclude my section, we have our 3-PGK test enzyme and its large domain movement, and we get this double spike. And then moving towards the biosensing part of this, we have two separate methods of detecting single molecule interaction of this interesting XG1 enzyme from the pathogen Candida albicans. So we can both use this for enzymology and biosensing, and we can actually see enzyme turnover at a single molecule level, which is really cool. So now we've got these interesting spectra. We need to actually figure out what this actually means. So I'm going to hand over to my colleague Jesus, who's going to take us through how we can use this data to extract some meaningful information.
I don't have too much time for my presentation. So Daniel, could you please next slide? I'm going to tell you about optoplasmonic sensing and femtonewton forces. Here is the sketch of our setup we used to measure our signals and to measure optical forces. It consists of tunable laser with collimating system and PDMS chamber with microsphere. PDMS chamber is filled with liquids under tests, And we excite whispering gallery modes inside microspheres with prism coupler. Here you can see the fundamental mode of the resonator. And also I try to show you a nanoparticle which is attached to the surface of microresonator. WGM microresonators have the highest quality factor. That means we have a very, very narrow spectral line, which allows us to detect single molecules. But the main disadvantage is they have very large mode volume because their sizes are roughly about 80 microns or even bigger. To minimize this problem, we can use gold nanoparticles or silver nanoparticles depending on their plasmon frequency. They provide us advantages for very strong mode localizations. Here I show that WGM resonators have quality factors more than 10 to the 6th, and at the same time, when we attach nanoparticles, they spoil quality factor of the systems, but the sensitivity of the system is increasing significantly. And also, we can use nanoparticles as sources of femtonewton forces. Next slide, please. So I don't have time. I think I was going to introduce optical tweezers in general, which were developed by Arthur Ashkin, who got Nobel Prize in 2018. Next slide, please. General concept of optical tweezers is just attraction particles, nano and microparticles with gradient forces which can be produced by intensity light, maybe milliwatt power of light. But they have conventional optical tweezers, they rely on the field gradients near the focus of a laser beam, which give rise to a trapping force towards the focus. The trapping volume of these tweezers is diffraction limited, and usually if we use lasers with 642 nanometers and 780 nanometers, this focus has the same sizes or the same order of sizes. The trapping volume of these tweezers is diffraction limited, so near field optical microscopy enables the optical measurements at dimensions beyond the diffraction limit. And the first nanotweezer was proposed in 1997 by Lucas Novotny and his colleagues, and it's just a metallic tip, gold tip with 5 nanometer tip radius, and here they numerically calculated optical trapping potential for sphere with a diameter of 10 nanometers and the electric permittivity 2.5. We also calculated how it could work for gold nanorod we used to detect our enzymes, and we modeled the systems using just small dielectric particle. And here you can see that roughly 10 femtonewton forces are quite achievable. Next slide, please. And I'm going to the experiment now. As you can see, femtonewton forces, which are trying to measure, they are proportional to the intensity of the incident beam. And if Daniel demonstrated results for beams with microwatts powers, I started to measure to make experiments on sensing of 3 PGK with huge power, 5.5 milliwatt power with the same resonators. Here, the main idea is when we apply very high power, maybe we will not be able to see any conformations. And when we reduce the power, we can just controllably change the molecule, controllably measure forces when we're trying to investigate conformational changes. And here is the protocol. I fabricated microspheres for this. I show how can we attach nanorods. Next slide, please. And this quite often happens we got unexpected results. We measured positive wavelength for a positive shift at full width at half maximum. And for the first time, we obtained a negative wavelength shifts upon binding of 3-PGK enzymes. Now we are in the process of explanation. Next slide, please. One of the possible explanations is based on physics. When we excite Whispering-Gallery modes, we have usually modes propagating in a clockwise direction. But when we add scatterers like our nanoparticles, we can also measure anti-clockwise modes which propagate in the opposite direction. And apparently, we measure usually one resonance, and we're looking for frequency shift. But in this case, when the power is quite big, apparently, counterpropagating modes is becoming weaker, and we can see negative shifts. But we still need to confirm this experiment. So next slide, please. And here is the conclusion. I think it's for Daniel.
So the kind of take-home messages is we can immobilize different enzymes onto a sensor, and we can actually detect single molecule turnover, particularly of this 3-PGK enzyme, and start to build a biosensor using this XG1 enzyme. As Jesus mentioned, we can combine this with information theory and actually start working out the dynamics of a single enzyme turnover, and but also starting to develop optical plasmonic tweezer system, so we can actually maybe even control the forces exerted by the enzymes. And just to acknowledge everyone in the project, particularly Simona, who did a lot of the 3-PGK work, and everyone else, and thank you for your attention. We have some questions as well. Yep, there we go.
Let me see if I can answer those.
I'm not sure.
Is that for me?
Analysts can
Okay,
actually
yeah.
answer them.
All right. Just checking whether I got everything.
Yeah, I do have the answers. I'll read them out in a bit when everyone's had a chance to have a go at them.
I see the question. I think here is one from, not sure from whom, but it says to Nikita, I'm not quite sure what your intentions were. By applying the forces, would they compress the enzyme? That's the question.
Thanks for the question. Yeah, that's what I mean. As you have seen, we have changes in the traces, and I suppose that we can increase the power when we're making experiments on sensing, and we can frozen to make it static. When we decrease the power, we can see them again, the conformational changes. That's the idea.
Okay, hopefully that answered the question. There's not much time for much more than one, so here's one from Abimbola. Have you worked on DNA binding proteins like ligase or restriction enzyme? I think that's for Daniel.
We have actually. The group has sort of a one of the first papers the group published was on a DNA polymerase enzyme. So I think we both immobilized the DNA on the sensor and then added the enzyme, and we did it the other way around as well and looked at what happened. So yeah, we have done that. That's quite an interesting field of research as well.
And last one, while we're on the minute, is the orientation of the nanorods defined, and are they added before or after adding the enzyme? Guess maybe Nikita then can answer that.
No, we cannot control it. It's just the binding events that what we can control with our techniques.
Yes, we're trying to go all
Are they
fabrication at some point.
after or before enzymes? Yeah, we fabricate microspheres, we attach nanorods, and after that several chemicals, and after that enzymes.
And maybe last one, does the immobilization step influence the activity of the enzyme for Daniel? That's the last question.
In a word, yes, it does. Sort of as a setup, we have to sort of think about which enzyme we want to use, because some enzymes don't like being immobilized to things. You might lock the active site. Interestingly, the XG1 enzyme becomes a lot more active when it's immobilized on things. So that's definitely a consideration we have to think about, and we're still sort of working on this to try and, I don't know, find out which method is the best one to use to actually immobilize an enzyme of your choice onto the gold.
Okay, with that, thanks to Jesus, Daniel, and Nikita. And
I should read out the answers for the.
Oh, okay, answers now.
Yeah. So number
We actually
one.
have answers.
Yeah, the answer's D. Number 2, C. Number 3 is A, and number 4 is D.
Didn't get that right.
I guess we hand back to Ollie.